Hi, good evening. So we will start today's session, which where we will be looking at week 10's content and then look at some of the example question answers. And this week's content is about nervous system. Okay, so I will start sharing my screen. All right. So let's begin the session. So in general, if we look at broadly what exactly is the nervous system, it is a highly organized network that actually allows us, not only us, in general living organisms, to sense their environment and to process the information that we obtain from the environment and accordingly respond to the stimulus or the information that we are receiving. Okay. Usually it operates using electrical impulses and there are multiple chemical transmitters or signals to rapidly transfer information across different parts of the body. And this is actually regulated by both voluntary actions like walking, speaking, etc., and involuntary actions like heartbeat, digestion, etc. Okay. Now, broadly, and also the human nervous system, the origin is ectodermal, and it is, if we divide it broadly into three parts, one is the CNS or central nervous system, then comes the which consists of the brain and the spinal cord. Then we have peripheral nervous system or the PNS, which actually comprises of the nerve systems arising from the cranium and the spinal cord. Okay, cranial nerves and spinal nerves. And then we have the autonomic nervous system, again divided into sympathetic and parasympathetic. So we will go through it one by one. In general, this is the complete nervous system of humans. Okay, and let's look at it one by one. So, central nervous system or CNS consists of the brain and the spinal cord. Now, if we talk about the brain, uh, it is actually residing inside the skull, the bony part, which actually protects and encapsulates the brain inside, whereas the spinal cord is actually enclosed by the vertebral column. And to support brain and to protect brain from external pressures, again, it is covered by a membrane of three layers of connective tissue called meninges, where the three layers are dura mater, arachnoid, and pia mater. Okay, and where this is the extra uh, outermost part, and the innermost layer is this pia mater, and this is the middle layer of the uh, triple connective tissue layer surrounding our brain. The innermost layer, which is the pia mater, it is actually thin and very delicate, and actually it has a lot of nerve endings. All right, nerve endings as well as definitely it there's a lot of blood supply as well. It is highly vascular. It is firmly adhered or attached to the brain, and then comes the middle layer, which is the arachnoid membrane, which is also thin, but it is highly folded and it is in front of the cranial venous sinus, especially a cavity. In general, when we talk about, when we mention the term sinus, that refers to cavities inside our skull. Okay. And the villi or the finger-like projections, the foldings of this middle layer or the arachnoid membrane actually look like villi or finger-like projections. It actually helps to reabsorb the cerebrospinal fluid or the CSF. Okay. So in general, the brain that we have inside, like encapsulated by the skull, it is actually surrounded by a layer of cerebrospinal fluid as well. The outermost layer, which is the dura mater, it is actually very thick, strong, and non-elastic, and it is made up of collagen fibers. And the space between the dura mater and the arachnoid is known as the subdural space, which is filled by a serous fluid. And the arachnoid, which is the middle layer, and the pia mater, the innermost layer, is known as the subarachnoid space. and it is filled by this particular fluid, which is the cerebrospinal fluid. Okay. Now again, when we talk about the forebrain or proencephalon, um, so it actually includes the cerebrum, diencephalon, and olfactory lobes. Now let us look at it one by one. Cerebrum is the largest and the most advanced part of the brain, where there are two cerebral hemispheres. Okay, and the two cerebral hemispheres are actually separated by a groove, which is known as the median fissure. All right, and both the hemispheres, the cerebral hemispheres, are connected by thick set of nerve fibers called as corpus callosum. Okay, and when we talk about the outer layer of the cerebrum, like outer portion, not really layer, the outer portion of the cerebrum is called the cerebral cortex, which again has numerous cell bodies and relatively few myelinated axons. Uh, so these are all parts of the neurons that we talk about. Neuron is the functional unit of a nervous system. Okay, so especially the cytoplasmic portion, which is called the cell body, and the long extended region coming out of the cell body is which eventually transmits the signals along like through it. So that is called as axons. Okay, and then there are endings and through which again neurotransmitters can come out, and eventually we can have a synapse, and then neurotransmitters can go into the target site. Like suppose we looked at the neuromuscular junction previously, right? So similarly, so these axons, these cell bodies, we will look at neurons in depth in later slides, but just because it is mentioned here, so mostly the cerebral cortex is made up of numerous cell bodies and relatively few myelinated axons. Also, these axons can have a layer of fats surrounding it, okay, a sheath kind of thing surrounding it, or it might not have the sheath surrounding the axon extension. Okay, so but however, if we talk about the cerebral cortical part, there we have, I mean, the cerebral cortex has numerous cell bodies. However, a few axon like extensions of neurons are present with having fewer layers of uh, that deposition, the sheath layer that we are talking about. Okay, so which eventually gives an overall gray appearance, and hence this part, the cerebral cortical part, is known as the gray matter. And the surface of the cortex is highly folded, and the upward folds are known as gyri, consecutive with the downward grooves, which are called sulci. Okay, or singular is gyrus and sulcus. So beneath the gray matter, there are actually millions of myelinated axon tracts and contain relatively few cell bodies. Okay, so especially the conducting region of the neuron is this portion is heavily populated in the layer beneath the cortex. Okay, however, the cortical region is actually populated by the cell body or the cytoplasmic portion of the neuron. Okay. So eventually the one that is the cell bodies, wherever the cell bodies are highly populated, that gives a gray appearance, and hence it is known as gray matter. Wherever in the region where the conducting region or the axonic part of the neurons are populated or crowded together, those those actually give a opaque white appearance, and hence that is known as white matter. Okay. Now, if we talk about the cerebral hemisphere, so we looked at that cerebrum is divided into two hemispheres, and each cerebral hemisphere is further divided into four lobes. Okay, where one is the frontal lobe here, or anterior one, exactly in the front part of the, like if we look at our skull or face, that is a frontal or anterior part. Then we have parietal or middle part. Then we have temporal or lateral part near the ears, and occipital or posterior lobe exactly behind, like the one connecting our neck towards that portion. Okay. So central sulcus separates frontal lobes from parietal lobes, and so these are all grooves. Okay, downward and upward grooves. So there are different kinds of grooves that are separating the different lobes. Okay, and when we talk about the different sensory areas, there are three main functional areas in each of the cerebral hemispheres. Okay, one is the sensory area, which is responsible, the region where which actually receives the impulses from receptors. Then there are association areas where the input signal is actually interpreted properly and stored as well, and eventually is actually analyzed, and then a response is given out. Okay, a signal is given out. So this area is actually, this association area in the cerebral hemispheres is actually involved in memory, learning, and reasoning. And then there's motor area, which actually is responsible for transmitting the output signal to the organs that will eventually give out or respond to the stimuli. Okay. And then we have diencephalon. We looked at cerebrum. Now we have diencephalon, which is again a small and posterior part of the forebrain covered by cerebrum. And this diencephalon has really important portions or parts. One is the thalamus, next is the hypothalamus, epithalamus, and metathalamus. Now let's look at it one by one. So thalamus is actually an important part of diencephalon, like really important part of diencephalon, and it represents the upper lateral wall. I mean, it is present in the upper lateral wall, and its function is to accept all the sensory impulses coming from all parts of the body except smell or olfaction, and send those to the cerebral cortex. Thus, it acts as a relay center to really kind of relay signal coming from one part of the body to another part for it to really process, or maybe pass on the signal to some other body part. Eventually, uh, like the concerned body part will be responding to the stimulus. Now, if we talk about lower animals, the thalamus actually acts as a sensory center because cerebral cortex is less developed there. Next comes hypothalamus. It is actually called the master gland. Okay, and it again represents the upper lateral wall was represented by thalamus. Hypo in general means lower. Okay, so something that is present lower to the, I mean, or beneath the or below the thalamus, it is called as hypothalamus. However, hypothalamus is the master gland, and it is present in the lower lateral wall of diencephalon. Then there are pituitary gland is actually attached to the middle part of the hypothalamus, and there's a web-like mesh which is found on the anterior surface of hypothalamus, which is known as the optic chiasma. And there is something called as corpus albicans found in the posterior part of hypothalamus. Now then comes the epithalamus, which is again some portion present on top of the thalamus. Epi means something on the outer or top portion. So it represents the roof of the diencephalon, covering the top part, and we have pineal glands present in this region. Then there's metathalamus, which actually represents the base or the floor of the diencephalon. Again, it has a medial geniculate body which is related to hearing, and another lateral geniculate body related to vision. Now, when we talk about the middle part of the brain, it is really small and contracted. And again, there are two longitudinal myelinated nerve fibers, which are, one is the cerebral pendicles, or located at the anterior part of midbrain, and then there are four spherical projections called optical lobe or colliculus, which are located at the posterior part of the midbrain. Okay. So then comes the hindbrain. Again, it is composed of different really important portions. One is the cerebellum. It is part of the hindbrain. Okay. It is the second largest part of the brain, the cerebellum, and it is made up of three lobes. Lateral lobes are really large and spherical, and it controls the regulation and coordination of voluntary muscles. In general, this with cerebellum, it also helps in maintaining the balance of a person, maintaining the balance of the body. Then comes medulla oblongata, which is tubular and cylindrical in shape and present at the posterior part. And it actually controls all, it is again a really important part which controls all the involuntary activities of the body, like respiration, metabolism, secretory actions of different cells, like that. And then we have pons. It is again a spherical projection situated below the midbrain, however, above the medulla oblongata, and it has many nerve fibers, okay, uh, like passing transversally or longitudinally. So transverse nerve fibers are they actually go and kind of converge with the cerebellum or join the cerebellum, whereas the longitudinal ones join the cerebellum to the medulla oblongata. Overall, this organ, this portion is actually helping in the breathing reaction through the pneumotaxic center. So midbrain, medulla oblongata, this part, and pons are situated on one axis, which is called, they together form the brain stem. So if we look at all these location and function of each regions of the brain, so if we talk about the prefrontal cortex, it is present at the frontal lobe and it is the site of intelligence, knowledge, and memory. Then we have the premotor area, which uh, has significance in which is the writing center, and even associated movement of it has implications or roles in the associated movement of eye, head, and body. It helps in controlling the complex movement of jaw, tongue, movements of jaw, tongue, pharynx, and larynx. Then there's motor area, which analyzes all types of voluntary muscle. Then we have the frontal eye field, which helps in the opening and closing of eyelid and helps in coordinating the movement of eye. Then we have Broca's area or motor speech area, which helps in speaking and analysis for like speaking and speech, basically. Then we have the auditory area, which is present in the temporal lobe, which analyzes the sound. Then we have olfactory area, which is again in the temporal lobe, uh, responsible for smell. And we have vernix area, which is also in the temporal lobe, analysis like region responsible for communication. Then we have the gustatory area, which is present at the parietal lobe, and it is responsible for taste. Somasthetic area, which is also present in the parietal region, which actually has implications in touch, pain, pressure, etc. Then there's angular gyrus, which is also the controlling center is in the parietal lobe, and it is associated with speech as well as in writing. And then we have occipital area, which is present in the occipital region towards the back, okay, and it has implications or roles in vision, okay. Now if we talk about the PNS, I mean this is sorry, this is part, I mean we talked about the brain, now we are talking about spinal cord only, which is part of CNS only, okay. Like central nervous system, we haven't yet come to PNS. So spinal cord is a continuation of medulla oblongata. We looked at this tubular part of the hindbrain, okay, the tubular part of the hindbrain that forms the brain stem together with midbrain and pons, okay. So it is a continuation, spinal cord is a continuation of this medulla oblongata, which comes out of again a region and a starting or beginning uh, portion which is called as the foramen of macrum and continues in the especially a cavity kind of area which eventually continues in the neural canal of vertebral column. It is also so spinal cord also has membranous covering. It is covered by dura mater, arachnoid, and pia mater, like we saw in the brain. And the narrow space between dura mater and vertebra is known as the epidural space. An outer part of spinal cord is of white matter, which is opposite to what we saw in the brain, right? We saw the outer cerebral cortex being mostly populated by cell bodies and less by myelinated axons, and which eventually gave it a gray appearance. However, immediately below the cortex, what we saw, it's mostly populated by myelinated axons, giving it a white opaque appearance. However, it is the opposite in case of spinal cord, where the outer part of spinal cord is made up of white matter, whereas the inner part is made up of gray matter. And eventually, the gray matter projects out into something called as into like forms the dorsal and ventral horns, and this eventually continues in a tube-like structure. Okay, and these together with uh, like tube-like structures together form the again at tube-like structures at the dorsal as well as the ventral portions, thereby forming the dorsal root ganglia. There are sensory neurons found in this particular region. This is a very important region, the dorsal root ganglia, where the axons extend and get embedded into gray matter of the spinal cord. And eventually, there are motor neurons as well, which are found in the ventral portion. We looked at the dorsal portion, right? So, which is also populated by sensory neurons, where eventually they make synapse with the ventral root neuron. Then there are motor neurons, which are found in the ventral root, where the cell body is found in the ventral horn, whereas the dendrons are embedded in the gray matter. So, spinal cord actually acts as a bridge between brain and different organs of the body. It regulates and conducts the reflex action as well as it provides the relay path. It is actually a relay center or maybe a middleman, okay, like a middleman for conducting the different kinds of impulses or different kinds of signals that eventually come from the brain. Now, if we talk about the reflex actions, so reflex actions are actually involuntary in nature, and it is very quick in nature, which is completed very quick, whereas as compared to normal actions. And again, there are these can be of two types. One is the cranial reflex, where the actions are actually completed by the brain, which are comparatively slow, like watering of mouth to upon seeing a good food, okay, or upon smelling a like really tasty food. And then there's spinal reflex, where the actions are completed by the spinal cord. These are really fast actions. For example, withdrawal of arm at the time of pinching by any needle, pricking by any needle, or if someone is pinching you, or maybe you touch a hot object, your feet or your hands touch a hot object, you immediately uh, pull it off, right? Pull it towards yourself, like pull it away from the uh, stimulus, causing the pain or causing the like giving the signal. Okay, so these are mostly spinal reflex. Now coming to the PNS or peripheral nervous system. So all the nerves arising from the brain and spinal cord are comprising of PNS or the peripheral nervous system. So the nerves which arise from the brain are termed as cranial nerves, whereas the nerves which arise from the spinal cord is termed as the spinal nerves. So overall, if we talk about reptiles, birds, and mammals, they all have 12 pairs of cranial nerves, whereas amphibians and fishes have only 10 pairs of cranial nerves. Now in humans, so these cranial nerves are numbered like 1, 2, 3, 4, like that. So in humans, the first, second, and the eighth cranial nerves are completely sensory in nature. They will only, uh, just uh, relay or transmit, like sense the stimulus and transmit the signal. That's it. They won't really participate in the response, okay, or the action in response to the signal received. And then there are cranial nerves numbered third, fourth, sixth, 11th, and 12th, which are motor, okay, motor nerves. And then remaining ones out of these 12 cranial nerves are actually mixed type. They can be sensory in nature sometimes, and they can also help in transmitting the signal and eventually causing a response. Okay, they are involved in both, like like the receiving the signal as well as responding to the signal received. Now in humans, there are 31. So we talked about cranial nerves here. Now, if we talk about spinal nerves, there are like 31 pairs of spinal nerves, and each spinal nerve is of a mixed type. Like it can be sensory as well as motor in nature, and it arises from, so each nerve arises from the roots of the horns that we talked about of gray matter of the spinal cord. Spinal cord, in case of spinal cord, the gray matter is towards the inside, white matter is outside. Uh, in like contrary to what we saw in the brain, where the outer region of the cortical region is composed of gray matter, and immediately beneath the gray matter, immediately next to the gray matter towards the inside, we have the white matter. Okay. So again, spinal nerves can be divided into 5 groups according to its position. Cranial spinal nerves, there are 8 pairs. So we have 31 pairs, right? So out of these, Cranial spinal nerves, 8 pairs. Thoracic spinal nerve, 12 pairs. Lumbar spinal nerve, 5 pairs. Sacral spinal nerves, 5 pairs. And coccygeal nerve, 5 pairs. So all of these are referring to the individual vertebral region, like different individual vertebra. Okay, that is enclosing these spinal nerves, and it is actually named based on those regions or those parts. Okay. So if we look at the different types of cranial nerves and their origin and distribution and function, so there's this olfactory nerve, which the source of it is epithelium, olfactory epithelium. It is sensory in nature and helps in smelling. There's optic nerve, origin is retina, and then it is sensory in nature, helps in vision. Then we have oculomotor and trochlear nerves, which arise from the midbrain, and again they are both are motor in nature, and the oculomotor one is involved in the movement of eyeballs, and trochlear one is involved in the rotation of eyeball. So both help in moving the eyeballs. Okay. Then we have trigeminal nerves, uh, again arising from pons varoli. One is ophthalmic in nature, maxillary and mandibular. So trigeminal in general is mixed in nature. So ophthalmic and maxillary are sensory, where okay, and ophthalmic ones are mostly present in the skin of nose, eyelid, forehead, scalp. Then there are lacrimal glands also, like the tear glands are also there. Okay, and then we have maxillary ones, which actually innervate the cheek area, upper lip, and lower eyelid. Then we have the mandibular ones, which innervate the lower jaw, lower lips, and ear region, especially the pinna of the ear. Okay. So then we have abducens nerve, again arising from pons, helps in rotation of eyeball. It is motor in nature. We have facial nerves, again arising from pons. It is mixed in nature. And then auditory, glossopharyngeal nerve, vagus nerve. Vagus nerve arises from medulla oblongata. It is mixed in nature, and it helps in, uh, transmitting or relaying the signals to larynx, lungs, heart, stomach, intestines. I mean, not only relaying the signal, like it is of mixed type, so it can receive as well as transmit the signal and lead to a corresponding response. And we have hypoglossal nerve, which again arises from medulla oblongata, which actually innervate the muscles of the tongue. It is motor in nature and helps in movement of tongue. So while I'm speaking, my hypoglossal nerve is in action. Okay, it is actually especially in terms of uh, the response. Okay, that I am giving out. While my words are coming out, so with the help of the hypoglossal nerve. I mean, there are different other nerves definitely. So the movement of tongue is being affected by hypoglossal nerve. Now, when we talk about autonomic nervous system, um, it actually controls the activities inside the body, which are mostly involuntary in nature, like heart beating, sweating, peristalsis, or the contractions and relaxations of the food pipe, etc. So it actually consists of motor neurons passing to the smooth muscle of internal organs, and it actually plays a very important role in maintaining homeostasis. Again, the autonomic system is divided into two parts, sympathetic and parasympathetic. Sympathetic is related with intuitive reaction, which increases the protection of the body in adverse atmospheric conditions, along with consumption of energy, whereas the parasympathetic ones are linked with the reactions where the energy is conserved. Okay. Now, how do we really measure nerve conduction? So, something called as nerve conduction study or NCS, it is actually a medical diagnostic test. What happens in nerve conduction study or NCS is the eventually the function and the capability of an electrical conduction is measured of the motor nerves as well as the sensory nerves. Okay. And the parameter that is used is the velocity, the nerve conduction velocity. So what happens in NCS is that it is also clubbed or integrated with electromyography, which measures the nerve and muscle functions together. Now, if there is any defect in the spinal nerve compression or any other neurologic disorder or injuries, these are actually undertaken for study by nerve conduction, like by NCS process. And eventually, the numbness of limbs, weakness of the legs, arms, as well as any tingling or burning sensation in particular areas of the body are usually, considered while diagnosing. And there are different kinds of nerve conduction study. Like one is the motor NCS. It actually, what happens in there is you electrically stimulate a peripheral nerve and then record the signals emanating, coming out of this particular stimulation from a muscle to which these nerve actually supplies the signal or the nerve endings are. Now, latency is something which is defined as the time taken for this electrical impulse to travel from the stimulating, like from travel from the time of stimulation to that particular, target of action, target site, which is a muscle in this case. And this particular latency is the time, and this time is usually in the range of milliseconds. Okay. Then there's sensory NCS, which is actually again similar to motor NCS, where you are actually stimulating electrically peripheral nerve, but you are actually recording it from a truly sensory portion of the nerve. Okay, which, as the name suggests, where sensory latencies are measured, which is again in milliseconds range. However, the sensory amplitudes are much smaller than the motor amplitudes. Okay, and sensory NCB is calculated in the same way as motor NCBs. Now, there is something called as F-wave study, where the motor and sensory segments are concerned about the nerve conduction velocity, the one that we talked about here, okay, which is the which is one of the very frequently used parameter during nerve conduction study. Now, what happens in there is, in case of F-wave study, you are actually trying to measure the nerve conduction velocities in sections or segments of your limbs, whereas where the F wave latency, again, another term is used to derive the conduction velocity of nerve between the limb and the spine. And what happens in this particular study, a strong electrical stimulus is actually given to distal portion of the nerve, and the resulting response or the impulse which travels in both directions, one can travel towards the muscle fiber and the other back to the motor neurons of the spinal cord. Then there is something called as H-reflex study, similar to the F-wave study, which evaluates the conduction between the limb and the spinal cord. Although there's a subtle difference where the impulses are going toward, like, based on the direction of the impulses. So where whenever the impulses are going towards the spinal cord, these are actually sensory in nature, whereas the impulses coming from the spinal cord are motor in nature. So how to really interpret? The interpretation is a complex, it's actually really complex, and expertise is required. Like, especially the expertise comes from neurologists, like physiatrists and neurophysiologists, which who are actually carrying out this particular study. And it has actually been really helpful in diagnosing many diseases related to nerves. And definitely it's non-invasive, and it can be sometimes painful because you are electrically stimulating the nerves, right? So you can have minor electrical shocks, but although the electrical current that is given for stimulation is actually low in amount and considered safe. So, however, there are patients who have devices like permanent pacemakers in their heart. So they are actually advised not to undertake this particular type of study. Then we have EEG or electroencephalograph. Okay. So what actually happened is way back in 1929, there was someone called Hans Berger who actually found some electrical activity when he connected a galvanometer to the human scalp. So eventually electroencephalography or EEG was born, which where an electrically operated instrument having attached to some 16 to 30 electrodes are attached to the scalp for a short time, which eventually gives electrical wave signals. And how this machine or instrument operates is that it detects the brain's electrical charge, and this electrical charge is actually maintained by numerous neurons. Okay, we talked about the functional unit of a nervous system, which is the neuron, and we will eventually read about it in like within few slides. So so how the neurons are charged? They are electrically charged by continuous pumping of ions by the membrane transport channels proteins across membrane transport proteins, and they constantly exchange ions with the extracellular matrix or fluid, for example, to maintain the resting membrane potential. When many ions having similar charge are pushed out of several neurons at the same time, they can push their neighbors who further apply force to their corresponding neighbors, and so on. Eventually, a wave is formed, and when this wave of ions reaches the electrodes attached to the scalp, this can actually sense, give, or take electrons from or from the metal or to the metal of the electrodes. And eventually, since the metal can conduct these electrons nicely, we can see voltage differences between any two electrodes, and eventually this the voltage difference is measured by a voltmeter. Now, there are certain kinds of wave patterns when we record an EEG. So, there are delta waves where the frequency is below 4 hertz, and it is high, it has the highest amplitude, and it is the slowest waves. It originates normally in adults during sleep, and it is also seen normally in babies. And eventually, these delta waves can actually also be observed in patients during coma. Then there are theta waves, whose frequencies range from 4 hertz to 7 hertz. So as the frequencies will rise, eventually the amplitude will go down. And theta waves are usually seen in young children. It can also be seen in older children and adults when they are under stress or during meditation. And uh, um, eventually, if there's excess theta activity at a certain age, then it represents abnormal activity. Then there are alpha waves, where the frequencies are much higher, ranging from 7 hertz to 14 hertz. So an awake person, but at rest, produces alpha wave. So Berger is termed this particular coined this term alpha wave when he saw the first rhythmic EEG activity. This was the posterior basic rhythm seen in the occipital regions of the brain. It arises with closing of the eyes and with relaxation, and weakens with eye opening or mental labor. In addition to the posterior basic rhythm, there are other normal alpha rhythms known as mu rhythm, which arises when hands and arms are indolent. Then there are beta waves, where the frequencies are again much higher, 15 hertz to 30 hertz. During extreme mental activity, beta wave initiates from the frontal and parietal regions, and it is closely linked to our motor behavior, and generally it weakens down during active movements. So an alert, wide awake person shows unsynchronized beta wave. Then there are gamma waves where the frequency is much really high, or probably the highest, which is 30 to 100 hertz. And the rhythms represent binding of different populations of neurons together into a network for the purpose of carrying a certain motor function. Then there are mu waves where the frequency is 8 to 13 hertz, partly overlaps with other frequencies, and denotes the synchronous firing of motor neurons in the resting state. So if there are deviations from the normal patterns, then it is an indication that there is some disorder, like there are some brain disorders, and some changes in the physiological state of the brain. And eventually, EEG recordings can actually help in diagnosing epilepsy, brain tumor, any kind of boils, or uh, in the brain, any kind of sleep disorders, metabolic or drug effects on the brain. Now, if we talk about neurons, so Neurons are the primary information processing. It is the functional unit of the nervous system. It actually processes and signals the cells of the, like these are the processing and signaling cells of the nervous system, and each of them can actually receive, conduct, and transmit electrochemical signals, which eventually helps in rapid and precise communication within the body. Now, if we talk about the structure of a neuron, it has three main parts. One is the dendrite, which are like branched extensions, which eventually receive signals from other neurons or sensory receptors. Then there's the cell body. We have been talking about gray matter and white matter. So this is the cell body or the soma, which contains the nucleus, like the cytoplasm, which contains a nucleus and organelles, and it helps in integrating the incoming signals. Then the long projection, which transmits impulses to other neurons, muscles, or glands. This is the axon. And the axon ends are covered by axon terminals, which release neurotransmitters into the synapse or the gaps between neurons or between a neuron and its target cell. How we looked at the neuromuscular junction, right? So in that case, the synaptic junction was neuromuscular, which was a small gap between neuron and muscle cell. Okay. Now, types of neurons can be sensory or afferent neurons, which carry information from the sensory receptors to the nervous system. And then there are motor or efferent neurons, which transmit instructions from the CNS to the effector cells, like muscles or glands. Then there are interneurons, which are found mainly in the CNS, which connects the sensory and motor neurons and perform the processing of the signals. And neurons do not divide in adulthood, but they can actually form new connections. There are few exceptions, but neurons can actually form new connections and modify their signaling strength through a process known as synaptic plasticity. So this is the structure of a neuron. I mean, this is how it schematic diagram of a neuron. This is the cell body or soma. This is the long projection, which is the axon. And these are the terminals, axon terminals, and these are the extensions from the cell body, which are called dendrites. Okay. So two parts, cell body, this one, or the cyton, and this cell process. Cyton is a broader part of neuron which contains uninucleated cytoplasm. That means only one nucleus is present. And except centrioles, all types of cell organelles are found in cytoplasm because the neurons do not divide, they do not have this centrioles present in there. Due to the absence of centrioles, neurons can't divide, and there are some other cell organelles like neurofibrils and Nissel's granules found in the cytoplasm, which helps in transferring the impulse to the cell body. And Nissel's granules are actually involved in protein synthesis. Okay, so Nissel's granules are formed by coiling of endoplasmic reticulum around the ribosome. So these are like Nissel bodies or Nissel granules, which are distributed throughout the cytoplasm of the cell body, except in the region close to the axon, this portion. Okay, these are actually mostly concentrated inside the cell body, but as we move towards the long projection or the axon area, the distribution lessens. Okay, close to the axon area. So the starting of the axon, I guess, the ending of the cell body and where the axon can actually project out, this region is called like the axon hillock. So Nissel granules are composed of rough endoplasmic reticulum and they are responsible for protein synthesis. And we have cell processes either towards the cell body or towards the end of axon. One is the dendrons or dendrites, this is the long axon, which are the cell processes of neuron. Fine branches are called dendrites, emanating the dendron, which contains some receptor points and it can receive the stimuli and produce centripetal conduction. Axon is the longest cell process of a neuron and it is covered by axolemma. Lemma, anywhere the term lemma comes up, lemma means layer. Okay. So the part where axon arises from the cell body is the axon hillock, and cytoplasm is known as axoplasm, which only contains neurofibrils, this one, okay, and mitochondria. And the terminal end, this one, is vesicular and called telodendria. Now there are some neurons which are covered by a layer of phospholipids, sphingomyelin, called as myelin sheath or medulla, like bundles of these ones. And myelin sheath is covered by a thin cell membrane which is called neurilemma or Schwann cells. So myelin sheath can act as an insulator and prevent leakage of ions across the axon. And based on how a neuron looks like, they can again be unipolar, bipolar, or multipolar. So in unipolar, you have a single process arising from the cell body. For example, in the nervous system of embryo, sometimes this single process can further divide into two processes. One of them can act as an axon, while the other one can act as a dendron or dendrite. And these kinds of neurons, the unipolar ones, are termed as the one with two processes are termed as pseudo-unipolar. Okay. And then there are bipolar neurons with properly distinct two processes arising from the cell body, where one is the axon at one end and dendrite and on the other end. And these types of neurons are actually found in eyes, the retina, okay, the olfactory epithelium, vestibular and cochlear ganglia again present in the ears. Okay. Then there are multipolar neurons. So they have one axon but many dendrons. So motor neurons and interneurons are multipolar in nature. Now, if we talk about hydra, neurons of hydra and a macrine cell of retina have no definite cell process. So these neurons are known as apolar neuron. Okay. So this is the diagrammatic representation of the same. Now, if we talk about the similarities, unipolar, bipolar, and multipolar neurons have some similar characteristics. They all feature long structures extending from the cell body. Additionally, they all have the main components of the cell body, axon and dendrite. Despite occurring in different areas within the body, the neurons play a similar role as they are responsible for passing the information between brain and CNS. Now, if we talk about differences, the number, it actually mainly differs in the number and structure of the components. For example, the dendrites only occur at the tips of the axons in unipolar neurons, but multiple dendrites stem or arise from the cell body of a multipolar neuron. And the structure of bipolar neurons is similar to that of pseudo-unipolar neurons in that two distinct structures stem from the cell body. However, instead of starting as one extension that splits into two, the structures grow independently out of the cell body. The structure of a multipolar neuron is different again as more than two structures extend from the cell body. Again, we were talking about afferent neurons and efferent neurons, right? So this is a diagrammatic representation of the same. Okay, like afferent neurons carry sensory info from receptors in skin or other organs. Well, it transmits it to the central nervous system, and cell bodies are located outside of the spinal cord in this case. So this is taking in this part. The region shown in blue is the afferent neuron. Then what you have here in purplish color or lavender color, it's autonomic division, which regulates involuntary body responses, blood vessels. It innervates the blood vessels. Okay. Then you have this red one here or the pink one here, which is the efferent neurons, which carries motor information from the brain to the peripheral nervous system. And cell bodies are located in the ventral horn of the spinal cord, and eventually it innervates the somatic division where the voluntary movement by skeletal muscles can take place. Like this is an example, just like diagrammatic example. So these are the different structural differences of afferent and efferent neurons. And there are two main properties of nervous tissue, which is excitability and conductivity. Excitability is the ability of nerve cell and nerve fiber to enter into a state of excitation in response to any kind of signal or stimulus. And the transmission of excitation in a particular direction is called conductivity. Now, this is a very important concept, which is the resting membrane potential. So, the electrical impulses that we were talking about. So, the cell membrane of a nerve cell is actually polarized in nature when negative potential exists more inside the cell as compared to the outside. So, this potential difference is actually approximately minus 65 millivolt across the cell membrane, which is at rest, okay, which is called the resting membrane potential. Now, the resting membrane potential, how it is maintained? It is maintained by an active transport which utilizes energy or ATP, active transport of ions against their electrochemical gradient by sodium potassium channels, which are proteins present at the membrane, and also by passive diffusion of ions. So, for active transport, there are carrier proteins located in the cell surface membrane, and they are driven by energy supplied by ATP and coupled by removal of three sodium ions from the axon with the help of uptake of two potassium ions. Okay. Three sodium ions uh, here and two potassium ions like in exchange of two potassium ions. So there's the passive diffusion of ions which opposes the active movement of ions. So what happens is the rate of diffusion depends on the permeability of the axon membrane for the ions. Now potassium ions actually have more permeability, so they can easily move through the membrane as compared to the sodium ions. Therefore, the loss of potassium ions is more than the gain of sodium ions. This leads to the net loss of potassium ions from the axon and generation of negative charge within the membrane. Now, what happens during an action potential or the excitation stage, which is also known as depolarization, which initiates a nerve impulse or spike, which is also called as active potential or action potential, generated by change in the sodium ion channel. Now, what is happening here? The sodium ion channels are voltage gated. At resting stage, these channels remain closed due to binding of calcium ions. When there is an action potential, it is generated by a sudden opening of the sodium gates, and opening of these gates increases the permeability of the membrane for sodium, which then enters inside by diffusion. This increases the positive ions inside the axon, and hence the negative potential inside the axon drops, and a change of minus 10 millivolt potential difference can be seen from resting membrane potential, sufficient to trigger a rapid influx of sodium ions, which leads to the generation of action potential. Now, eventually what is happening is, first the negative resting potential is being cancelled out due to influx of a lot more amount of sodium ions. At this point, the membrane gets completely depolarized and the potential difference is formed across the membrane. So this difference is at 30 millivolts, which corresponds to the maximum concentration of sodium inside the axon. Now what happens during repolarization? A fraction of a second after the sodium gates open, membrane gets depolarized. Potassium gates can also open eventually, and hence the potassium is diffused out of the axon. Again, this causes less positive charge inside with respect to outside, and thus due to repolarization, the potential again changes from 30 millivolts to minus 65 millivolts. Now the neuron is prepared for receiving another stimulus and to conduct them. So it's necessary to actually restore this normal resting potential by expelling out the sodium ions and taking inside potassium ions. This time that is taken for restoration is called refractory period, because during this period the membrane can't actually receive another impulse. Now when we talk about the synapse and the synaptic junction, we looked at it earlier also. This is the area of functional contact between two neurons. In a synapse, membrane of telodendria, the ending of the axons, is called as presynaptic membrane. Here, this one, presynaptic membrane, which is passing on the things, and then the membrane of the dendron of the other neuron, which is receiving the signal, is known as the postsynaptic membrane. Okay, and the space between these two membranes is called a cleft, synaptic cleft. Okay, we looked at acetylcholine receptor and acetylcholine neurotransmitters in the neuromuscular junction earlier as well. Now let us look at some of the questions. So which statement about synapses is correct? Electrical synapses are slower than chemical synapses. Chemical synapses are unidirectional. Electrical synapses require neurotransmitters. Chemical synapses allow direct ion flow. So it is talking about the correct statement, which is this portion. Option B, that chemical synapses are actually unidirectional. And electrical synapses are actually much faster than chemical synapses, and they allow direct ion flow via gap junctions. Now, a patient cannot form new memories, but the patient can remember the old ones. So, which region is likely damaged? So, the region is the hippocampal region, which is essential for consolidation of memory. Now, which structure produces the cerebrospinal fluid? So, these are the different membranes, the triple membrane structure of the brain as well as the spinal cord. Now, which particular portion is producing the cerebrospinal fluid? This is the choroid plexus, which are present in the brain ventricles. Okay. Now, there's a toxin which is blocking the voltage gated sodium ion channels. So, what will be the immediate effect? Now, if voltage gated sodium ion channels are blocked, the immediate effect will be no action potential generation, because that is how the very first depolarization is taking place due to sudden opening or influx of the sodium ions inside the axonic membrane. Okay, so without sodium ion influx, action potential cannot be generated, and hence the nerve transmission will be stopped. Now there are two statements. One is the myelin sheath increases conduction velocity, and next is the myelin increases membrane capacitance. So the correct option will be myelin actually decreases the capacitance and increases the resistance, because it is a lipid layer, like lipid covering on the axon, which actually is not very um, conductive for, like, it's not really a good conductor of the electrical impulses. So eventually it is increasing the resistance, and hence there's faster conduction, because so if there are, like, suppose this is a there is huh. So this is covered by the myelin sheath. Okay. Now these are the areas where the axon terminals are there. So if there is an impulse, because this is covered by fat layer, okay, lipid layer, what happens is that there is jumping conduction, which is also known as saltatory conduction. So this is much faster, because there is this lipid thing, it can't really pass through this area. So it actually jumps from here. So there is saltatory conduction of nerve impulse. So here is the question. Yes. So myelin sheath increases conduction velocity and myelin increases membrane capacitance. So the first one is true. I mean, the first statement is true. Okay. But it actually isn't increasing the membrane capacitance. It is decreasing the membrane capacitance and increasing the resistance, because it is bound by lipid layer. Now, in the reflex arc, the ones that we were talking about, it can be either spinal or cranial. The correct sequence is you receive something, then it has to be sensed, and there should be some kind of mixing and association and integration. Eventually, it is passed on by the motor neuron, and there is effected. Okay. So, the correct option is this is the standard reflex arc pathway, this one, which ensures rapid response without conscious brain involvement. So, everything is happening in the, like, with respect to the spinal cord. Okay. Now, which step is calcium dependent in synaptic transmission? So, the thing is vesicular fusion with presynaptic membrane. So, calcium influx actually triggers a synaptic vesicle fusion, and hence it releases the neurotransmitters inside the synaptic cleft. What happens if there is damage to the cerebellum? Cerebellum is associated with um, coordination. Okay. So, you can have ataxia, which actually helps in movement coordination. And if there is any damage to cerebellum, then you can have damage to, like, you can have loss of coordination or ataxia. If we talk about the hypothalamus, it is the master gland. It is primarily responsible for this homeostasis and endocrine control. It actually regulates temperature, hunger, thirst, circadian rhythms, and controls the pituitary gland as well. Now, which neurotransmitter is primarily inhibitory in the central nervous system? It is GABA or gamma aminobutyric acid, which actually opens the chloride channels, leading to hyperpolarization, eventually leading to an inhibitory effect. Now, during the rising phase of an action potential, which ion movement predominates? If it is rising, then we are actually having a lot of influx of sodium ions. Okay, so this rapid depolarization is due to opening of the voltage gated ion channels, sodium ion channels, which leads to rapid sodium ion influx. So the correct option is option B. Now, which of the following correctly describes saltatory conduction that I was talking about? That is jumping of action potential between nodes of Ranvier. So the areas we saw, right, the blobs of sheath, and then there are gaps in between. So those are the nodes of Ranvier on a myelinated axon. So correct option is option B. Saltatory conduction occurs in myelinated neurons where the impulses can jump between the nodes of Ranvier, which increases the speed and efficiency. Now, a patient touches a hot object but does not feel the pain. However, they can still move their hand voluntarily. So, which particular neuron type is most likely damaged? So, the neuron type that is damaged is the sensory neurons, because the person is touching the hot object but doesn't feel the pain, but they can move the hand voluntarily. So, they are not eventually sensing the stimulus here. So the problematic portion is the afferent or sensory neurons here. Okay, because movement or the response or the signal, output signal is actually intact. So the different pathway is normal. However, the sensory pathway has been affected. Now, the patient cannot move their limbs but can feel touch or feel the pain. So this is the complete opposite to the question stated above. So which neuron is affected? Then the motor function is affected where the response is not there, but one can eventually sense the stimulus. Okay, since the sensation is intact, so afferent neurons are working fine. However, the response is not there, so there is some issue with the efferent neurons. All right. Now, a patient loses reflex action in a limb. Testing reveals sensory neurons are actually intact, but there's no motor response. So, which component is most likely damaged? If there's no motor response, then the efferent neuron is most likely damaged here. Okay. Now, a neuron has a single process that splits into two branches, one towards the periphery and one towards the CNS. So, which is the neuron type here? So the neuron type is, we looked at it, it is pseudo-unipolar, which is common in dorsal root ganglia, and eventually it bifurcates. Only one process can bifurcate and giving rise to pseudo-unipolar neurons. All right, that's it for today's session. We will again meet tomorrow actually with week 11 content, and we will look at week 11's question answers related to week 11 content. Okay. So we'll be meeting tomorrow again at 3 p.m., 3 to 5. And so thank you for joining today. See you tomorrow. So I will stop presenting and